Your neighbor. Yeah. Yeah, she went to. What I have here. Buenas tardes, le doy la bienvenida a todos y todos. Antes de iniciar, les recordamos que las puertas en ambos lados del auditorio son salidas de emergencia, por lo que hay que dejar las pasillas libres. Si suena una alarma, usa la salida más cercana, hay una punta de reunión atrás. Queremos otra vez dar las gracias al posgrado en Ciencias Biológicas y la Coordinación de Investigación Científica por financiamiento, por traer a los ponentes para este evento. Muchas gracias. Y hoy es un placer presentarles a la doctora Emily Sessa. La doctora Sessa es la directora del Herbario William y Linda Steer del Jardín Botánico de Nueva York. Es botánica y sistemática de plantas y su investigación se centra en la ecología y evolución de los helechos y las licofitas. Obtuvo su licenciatura en Ecología y Biología Evolutiva en la Universidad de Cornell en 2005 y su doctorado en Botánica en la Universidad de Wisconsin-Madison en 2012. Después de un año de formación postdoctoral en Genómica de Plantas en la Universidad de Arizona, se unió a la Facultad del Departamento de Biología en la Universidad de Florida en 2013, donde obtuvo definitividad en 2019. En agosto de 2022, se regresó a Nueva York para unirse al equipo del Jardín Botánico de Nueva York. La doctora Sessa continúa realizando investigaciones sobre la sistemática de los helechos, la biogeografía histórica y las respuestas al cambio ambiental. Ha publicado 82 artículos revisados por pares y un libro. Su investigación ha sido apoyada por la National Science Foundation y la NASA de los Estados Unidos. Les vamos a dar la bienvenida a la doctora.
Thank you so much, everyone. Thank you for coming back in the afternoon after you've all had lunch. And thank you so much for the invitation to be here. It's really an incredible honor. And this is my first time ever visiting Mexico. So yeah, thank you. This has been a really wonderful and very joyful place to be this week. And I wish I could stay and not have to go back to what awaits us back in the United States. So I'm going to continue our sort of organismal theme that we've had today. We've had two talks focused on different groups of organisms, and I am going to be speaking about the plants that are very close to my heart, which is the ferns. So we'll see if, there we go. So ferns are very beautiful. This slide is meant to give you a small snapshot of what fern diversity looks like. And one of the reasons I actually became interested in this group in the first place was that I found them very beautiful. They also are very interesting for a number of evolutionary reasons. They have very large genomes. They have some interesting evolutionary and ecological patterns, and they're very important parts of ecosystems. Often, I think, you know, if you may be familiar with the term plant blindness, that often people don't see plants when they look out in habitats or in forests, for example, but I always see the ferns, and they play very important ecological roles. So this is an example of a hammock ecosystem in the state of Florida, where everything in the understory basically is ferns. Another few examples, this is Hawaii, where this is a relatively recent lava flow on the big island of Hawaii, and all of the, or many of the initial colonists of this new substrate are ferns. They can grow in deserts and very dry environments. They can also grow in water. So this little floating island you see here, the really bright green color, that's actually all a fern growing there. So they play a number of different ecological roles. They can be found in many, many different habitats and ecosystems. So they're really very important plants. Thinking about sort of the taxonomy and classification of ferns, according to the most recent taxonomy, which was actually the first sort of broad scale classification system for ferns, the PPG, this was the fern and lycophyte equivalent of the APG system, and it was only published in 2016, so we were a little bit behind the flowering plant community. But there are about 10,500 species of ferns in about 320 genera, almost 50 families, and 11 orders, and they make up this class of Polypodiopsida. So with this number of species, they're actually the second largest group of vascular plants after the flowering plants, because of course the lycophytes and the gymnosperms have many fewer species. So this is nowhere near as many species as angiosperms, but they are technically the second largest group of vascular plants, vascular land plants. So my talk today is going to be in sort of three pieces. I'm going to tell you a little bit about some background on the history of fern phylogenetics, and I'll use that to kind of introduce some of the major groups of ferns. And then I'm going to tell you a bit about this project called GoFlag, which was a very large project funded by our National Science Foundation in the United States that is much broader than just ferns. But then at the end, I'll tell you about the results that we have from that project that are just about ferns. So for this first part, thinking about background of fern phylogenetics, I wanted to start by kind of describing what have been some of the earliest questions in fern phylogenetics. So you can't see the small text here, don't worry about that. But what this is showing you is a phylogeny of the ferns, and they are the sister group to the seed plants. So that is the seed plants right there. So ferns are sister to that group of the angiosperms and gymnosperms. And then within the ferns, there are three sort of two clades and then a set of lineages. So the first is this big group, which is called the leptosporangiates. And I'm showing it as one big clade here, but historically it was unclear whether this group was a clade or not. And that term, leptosporangiates, refers to the anatomy of the sporangium. So ferns are spore dispersing organisms, and the sporangium, this organ that disperses the spores, has a very unique structure in the leptosporangiates. But despite the fact that all of these groups share that structure, it wasn't clear whether or not they formed a monophyletic group. Then there are three groups that are called the eusporangiate lineages, and then there's another one that's the horsetails. And so I'm going to kind of blow up this section so that you can see it a little bit better. So here you see just the top of the leptosporangiates, and then there are these three at the level of order and family. These three groups make up the eusporangiates, and then here we have the horsetails. So I'm going to say a little bit more about each of these and show you some pictures to help introduce these groups better. So the leptosporangiates, this is the vast majority of the ferns. So well over 10,000 species, huge amount of diversity, different ranges of habitats, globally distributed, of course. So this is a really, really large group that we're talking about. So a number of orders, families, genera, and many, many species. And then the eusporangiates are these three distinct lineages, and the relationships among those three lineages have been complicated and difficult to figure out how those three groups are related to each other, and then how they're related to the leptosporangiates and also to the horsetails. So these eusporangiate lineages are also very different from each other morphologically, like very different. So the Maradiales, these are often tree-sized. They can be quite large. This over here is Psilotum nudum. This is the whisk fern. So this is a plant that for a long time was actually thought to be sort of a direct representative of the earliest land plants because it doesn't have true roots or true leaves, and actually homologies between its structures with roots and leaves remain a bit unclear. And then this is a representative here of the ophioglossales, which hopefully maybe can make out here that these little stalk-like structures, those are actually the fertile parts of the leaves, and then these are the sterile parts of the leaves. So a very sort of different leaf structure and different reproductive structure. So morphologically, these three groups are very different from each other. And then last is the horsetails, which in this phylogeny here are shown as sister to all of the other ferns. And horsetails are very beautiful. As you can see here, they also are quite different morphologically. So everything that you see in this picture, for example, that's green, those are actually branches. And this picture here in the center, these structures are the leaves. They're extremely reduced. And then here you have the spores arranged in these terminal strobili. So again, very different morphologically from any of these other groups that I've just shown you that are sort of candidates for being sister to the leptosporangiates. So figuring out how all of these things were related to each other at a very deep level in fern phylogeny has been of interest for a very long time and challenging given the morphological differences among these groups. So in the early 90s, up to about 2001, early 2000s, we started to get molecular data in the form of chloroplast restriction site maps and then the first chloroplast sequence data. So these are some of those papers, which these are like the Bible for fern people. These are very important early papers for us. So they were able to pretty confidently confirm that the leptosporangiates are a clade. So that was good. But on these other two questions, they were sort of equivocal. We didn't really have a clear answer to either of these questions from these early chloroplast-based studies. And I really like, this is a figure from one of Kathleen Pryor's papers in 2004. So she's showing the leptosporangiates as a clade here, but then you can see that there's some polytomy there. So lack of resolution of, in that case, the horsetails and the meradioids. So still a lot of work to be done at that point. So from then until now, roughly 20-something years, these have continued to be themes or big questions that we've had in the fern systematics community. So increasingly, as nuclear genomic data have become available, we're interested in what the plastid and the nuclear data have to say relative to one another. There are what I'm referring to as continually recalcitrant nodes. So nodes that no matter how much data we throw at them, they still seem to be difficult to resolve. And then, of course, increasingly, we're interested in things like molecular dating, looking at rates of evolution in different groups, and other sort of add-ons that you can do with phylogeny. So here's our plastid versus nuclear, our nodes I'm illustrating here with some polytomy, some lack of resolution, and then a fanciful way of showing some dates and rates. So today I'm going to be talking about this kind of comparison. We'll be talking about a number of those historically difficult to resolve nodes, and then I'll be talking about molecular dating at the very end and what we know now about dates for ferns. So this table, I don't expect you to remember all this information, but this basically is all of the previous, and Go flag is in there as well. So that's what I'm talking about today in green there and in blue there. So these are all of the previous sort of large scale studies on fern phylogenetics. And what I mean by large scale is that they had sufficient taxon sampling to be able to get at some of these questions, especially that involve the eusporangiates and the horsetails. So of course, there have been many, many other phylogenetic analyses that focus on individual families or individual genera, but this is really a comprehensive list of all of the really big studies on ferns. And they're organized with the green ones are all plastid. There's one mitochondrial-based phylogenetic study and then a number of nuclear studies. And you can see that the dates here they're arranged in date order up from 2004 to the present. The nuclear studies, the first one was 2015 up to the present. And then you can see that in general, the numbers of loci increase over time. And the number of taxa more or less increase over time, although it's not necessarily a linear relationship. And I'm going to be using this a lot later in the course of the talk. So I just wanted to introduce this quickly. And so the way that I'm going to use those papers, I want to give you a little toy illustration or example here, and I'm going to show you a lot more like this later. So I wanted to sort of give you a preview and let you see more of what's coming later. So This little table here, the set of boxes matches the set of boxes down here. And so what these little topologies are showing you are three possible resolutions of the relationship among these tips. So equisetales, other ferns, and seed plants. And so you can see there's three possible ways that those can resolve in a phylogeny. And if the squares were colored in like this, it would mean all of those papers found this topology. Does that make sense? Okay. If it looked like this, according to the coloring, that would mean all of the plastid phylogenies matched this resolution. And then the nuclear trees were split, where two of those studies found this topology and two of the studies found that topology. Good? Okay, good. We're going to see a lot more like this later. So you'll get more practice at looking at these figures later on. So next I want to switch to talk about GoFlag a little bit. So GoFlag, this was funded by a program through our National Science Foundation in the US called the Genealogy of Life. So Go Life, we called ours GoFlag for a reason I'll explain in a moment. And basically the goal of this funding program was to illuminate the dark parts of the tree of life. So NSF felt that it had been funding maybe relatively a lot of studies on parts of the tree of life that were sort of well understood, and they wanted specifically to target or encourage people to submit funding applications for parts of the tree of life that you could make a good case had been less well studied. And so we put together this consortium of faculty. Most of us were at the University of Florida, but then we also had some colleagues at the Field Museum in Chicago and at the University of Arizona. And we put together this grant called GoFlag that was focused on the flagellate plants. So here's a phylogeny of the land plants and an algal relative. And for these plants, flagellated sperm are the ancestral condition for green plants. And so here's some pictures of the beautiful flagellated sperm that you see in these groups. So these are some algal lineages. Anoclea and Marsilia are ferns. This is a cycad, and here we've got a liverwort. So you can see lovely flagella on all of these in varying sizes and orientations and arrangements. And then, of course, once you get to angiosperms up here, those little sperm there have lost their flagella, right? So these groups represent the flagellate plants. And a quick note about gymnosperms for the plant people in the room. So we wanted to focus on this set of plants with flagellated sperm. And the gymnosperms are interesting because within the gymnosperms, cycads and ginkgo do actually have flagellated sperm. The other gymnosperm lineages, the neophytes and the conifers, don't. But we included them as honorary flagellate plants. We wanted to be inclusive, and we weren't going to leave out some of the gymnosperms. So we included all of the gymnosperms, but it's important to note that they don't actually all have flagellate sperm. So why focus on this set of groups? What was our rationale? We convinced the National Science Foundation that it was worth focusing on this set of species. So what was our rationale for that? And our argument was basically that for the first 300 million years of plant life on land, it was the ancestors of our modern flagellate plants. That was what existed up until the evolution of angiosperms, of course. And within that time period, there were so many important innovations in plant evolution that to understand their evolution, you really need a comprehensive phylogeny for this set of organisms. So that included things like the movement onto land, kind of important. Various fungal symbioses that we now see in all of these groups that we understand are essential for their function would have evolved in the ancestors of these plants. Stomata evolved at some point I'm a little equivocal there because some of the bryophytes have stomata-like structures. Vascular tissue, true roots, true stems, really important structures, obviously. The shift to a sporophyte dominant life cycle would have happened somewhere along the route leading to the tracheophytes. Leaves evolved at least once, probably multiple times in different groups. Seeds, of course, would have evolved in the shared ancestor of the angiosperms and the gymnosperms, and secondary growth in that group as well. And then we have 3 different evolutions of heterospory. So homospory is the ancestral condition. Heterospory is a prerequisite for seeds, for example. So it's quite important. And the fact that it evolves multiple times, very interesting. In addition to those things you can map on the tree, there's also first transitions back to various types of aquatic habitats. It's quite important. Parasitism evolves. epiphytism evolves in the ancestors of these groups. The independent diploid sporophyte generation sort of goes along with some of the early evolution of life cycles back here. And then things like animal-mediated relationships that obviously are very important, and pollen and pollination as well. So these are really important plants to understand, I would still argue today. So GoFlag, I wanted to just briefly talk about the methods and what we did, but I'm not going to talk about this too much. If you have questions, I'd be happy to answer them later, or I'll refer you to the publication that came out a few years ago. So to develop this probe set, we used all of the available genomic and transcriptomic data. We relied heavily on the 1KP project. Thank you very much, Jim. It was very important for us. I have 1173 there. I can't remember if that's actually the number you had, close enough. And there were 241 flagellate land plants in the 1KP data. We added another 23 land plant genomes for these plants. And then basically extracted 410 single or low copy nuclear genes. We identified exons in those genes that were at least 120 base pairs long, and they had to have 65% pairwise identity across multiple of the genomes, and that had to be across at least 6 of those 23 genomes. Because we really wanted, this is obviously, like I said, 300 million years of evolution. We wanted to try to get loci that we could sequence across all of these. There were, we didn't want to, there's obviously a big danger ending up with things that only would work for ferns or only would work for liverworts, and we didn't want that. So we wanted to try to get things that would be representative across the group. So we ended up with 451 loci, so in exons. We did a pilot study to see how they performed, and then we did further optimization after that pilot study. And for the pilot study, our sampling included 188 flagellate land plants, a bit unevenly distributed. So 14 hornworts, 16 lycophytes, and 16 gymnosperms, and then more liverworts, mosses, and ferns. So 46, 48, and 48. And that did represent the bias of the investigators, to be honest. But decent sampling across all of these groups. So after we tested the probe set, the 451 probes, the probes for those loci with that group, we did a bit more refinement, and we ended up with what we called the GoFlag 408 probe set. So this was 52,000 some probes in 408 loci across 229 genes. So a gene could have, multiple loci targeted in multiple of its exons, basically. So this is the publication where those methods were published. It came out in 2021. So actually, I'll leave that up there if anybody wants to make a note of that, and I'll take a sip of water. And so I'll just mention some of the results that we published in that paper, just because they're of interest. So this is the distribution of loci that we were able to successfully sequence across taxa. And this is actually, this and the next slide are from the 451 probes. So it's number of loci binned by the number of taxa. So basically what this means is that there were, I think it's 16 species where there were less than 44 loci sequenced. But then, there's quite a large number over here where we're getting 300 or more loci successfully sequenced. And you can see the distribution across the different groups. So the number of loci recovered ranged from 3 to 436. So there wasn't a single sample that we got all 451, but the largest was 436. An average of 332, median of 394. And then fewer than 10% of the loci were recovered in only 16 samples. So that's good. And then in 82 samples, we got more than 90%. So that's this big bar here. So we were pretty pleased. We felt like we were getting a lot of the loci recovered, and certainly enough for robust phylogenetic analyses. And then this figure is showing you a couple of different ways of looking at our success in recovering loci based on either the amount of DNA or the numbers of read pairs that we had. So the thing to notice in this one over here is that we got a large number of loci recovered with a relatively small number of reads. And then over here, it's a bit more scattered, but you get a lot of loci pretty much across the board, regardless of how much DNA you had. And with the ferns, they were a little bit difficult, which I'm used to. So in some cases, even with a lot of DNA, we recovered fewer loci than we would have liked to, but still, up to 100 loci in some cases. And I'll also mention that we had very good success with herbarium samples, even quite old herbarium samples, which yielded relatively little DNA. We were actually able to get quite a lot of loci sequenced, which was a surprise and very happy surprise. So GoFlag, our vision when we started this project was that we wanted to transform the landscape of phylogenetic analyses for these plants. We wanted to generate an enormous amount of data, make it publicly available, and just provide far more genetic resources than had been available. And I think that we've achieved that. So if you do a Google Scholar search for citations of that paper, as of the other day, there were 70, I think, and it was only published in 2021. So there have been many, many papers, many, many talks. So they range from family level studies. So there have been a number of those, particularly in ferns. There have been some sort of genus level studies. So this one was on Physidens. This is Centrichia, and this is those are both bryophytes, and then the fern genus Polypodium. And these are just examples. This study of a new hybrid used GoFlag data to determine that that was a hybrid. And then if you may have seen, this very large paper on bryophytes was published earlier this year, I think, and that was entirely using GoFlag data. So it's been a very impactful project. And our model was that we basically, when we got the funding, we put the call out to the community and we basically said, we have money to sequence these plants. If you have samples, send us your samples. We will sequence them for you for free because we have the money, and then we'll give you the data. And eventually we want the data to be made publicly available, but you have first shot at publishing whatever you want to with your own data. So that was a sort of crowd-funded model, and we especially wanted to fund students. We wanted to get students' samples sequenced who might not have been able to get that done financially or for whatever reason otherwise. So now to the best part, which is the fern part. So we included 986 ferns, and obviously lots of outgroups from lycophytes. And we did also have seed plant outgroups in there as well. And the matrix that resulted for ferns. So I'm not gonna go in depth into our phylogenetic methods. I'm just gonna tell you a bit about the data set and then go straight to the results. But the matrix length was about 77,000 nucleotide sites, and a third of that for amino acids. And we had very good taxon occupancy. So on average, we had about 203 genes covered out of the 229. And again, with multiple loci, spread across those genes. And we concatenated loci within a gene, but then analyzed all of the genes separately. So we did a lot of, this will all be in the paper. I'm not going to talk about it too much, but we did a lot of sort of exploration of things like substitution models and how they affected our gene tree reconstruction. So we looked at models for nucleotides and codons and amino acids. And today I'm only going to show you results using the Astral species tree that used the best model for each gene tree. So whichever of those models was the best one for each gene, those were the ones that we took and put into Astral to make our species tree. We also were able to get a lot of off-target reads from which we could assemble plastid data. And so we ended up with 79 loci from the plastid genome, which was pretty good. So I'll also be telling you about those results as well. So this is what that nuclear species tree looks like. So this is the Astral tree, and all of the branches had maximal support. So these are local posterior probabilities for the primary topology. And you can see that, not surprisingly, where there are short branches, there tends to be a number, which means that it wasn't 100%. But even those numbers tend to be pretty high with a couple of exceptions in that part of the tree. These pie charts show you the quartet support. So again, this is the species tree. So that's the main topology, the primary topology. And then for every node, there are going to be 2 alternate topologies. So yeah, for any set of those number of taxa branching from a node, you're going to have 3 possibilities. And so you can kind of see anywhere the branches are long, you get pretty good support for the primary topology, but then where the branches are shorter, it gets a little more equivocal. So you have additional support kind of creeping in there for the alternate topologies. And these nodes here, or these pie charts here, these are showing the support for all of these individual nodes along the backbone. And then what these over here are showing you is the support for the crown of each of those families. Does that make sense? We're good? Okay. Cool. And so you can see the families are for the most part, they're quite well supported, or orders, I'm sorry. In some cases it's families, in most cases it's orders. So pretty good support. So the next one I'm going to show you is the nuclear and the plastid facing each other in a very friendly way, except for a few places I've highlighted where they disagree. So again, on this side in blue, so this is the nuclear tree, which is the species tree from Astral. And then for the plastid data, we concatenated all of the plastid loci, and that's an analysis done with IQ tree, which is a likelihood-based method. And here, the stars are showing you the nodes that had less than 100% support. So for the nuclear tree, it's local posterior probabilities. And then for the plastid tree, it's bootstrap and SH likelihood numbers. And so I've highlighted here in the center the places, and along the backbone in black, the places where the two trees disagree. So you can see there's sort of three main places of disagreement between the nuclear and the plastid phylogenies. If we think about these trees in sort of the context of the history of difficult areas of the fern tree, unsurprisingly, these three line up with sort of the difficult to resolve areas. This up here has also been a historically difficult to understand part of the tree. So I'm going to talk about that one as well. So we're going to kind of walk through these in an order of sort of less complex to more complex. So I'm going to bring back in the sort of colored in figure that corresponds to those papers. So you're going to see this little structure a lot over the next few slides. So the most important thing to remember is that all of these up here are plastid. The pink one is mitochondrial, and then all the blue ones are the nuclear studies. And they're sort of arranged from the oldest here, and then it picks back up here to most recent. So that's GoFlag, and then GoFlag is down there as well. And just again, to just sort of remind you very quickly of the rough numbers, most of those plastid studies had very few loci, varying numbers of species or of taxa sampled. And then these ones, there were a few that are basically whole plastomes, again, with varying numbers of taxa. And then on the nuclear side, most of them are transcriptomes. The GoFlag is these 408 low copy, and then varying numbers of species, with GoFlag by far the largest sampling of all the nuclear studies. Okay, so let's start with this one. This is the easiest one. So Cyatheales and Salviniales, these two, and they're resolving in different positions on these two trees. So the question here is essentially which of these groups, or how are these two groups related to this large group up here, which is the Polypodiales. And if you know anything about ferns, you know that's a really big group, because this group, I'm showing these triangles all the same size, but the Eupolypods is an enormous group. So these are each sort of smallish groups leading up to that really big group. And who the sister is to this set of families and orders is an important thing that we want to know. And they are very, very different, our two candidates here. So the Cyatheales are the tree ferns. The Salviniales are the water ferns. Really different. And there's not a lot of morphological information that we could use to help us figure out what's going on here. This group, I will also point out, is the one place in ferns where heterospory has evolved. So like very interesting for us to try to figure out what's going on here. So among all the data sets, these are the two resolutions that we see. So I'm not going to show you all the possible resolutions. I'm just going to show you the resolutions that we see in all these data sets. So for here, these are the options we see. So it's either that the tree ferns are sister to the Polypodiales, and then the water ferns are sister to those two together, or the tree ferns and the water ferns forming a clade that is sister to the Polypodiales. And as you can see, the vast majority of these studies support this resolution with the tree ferns sister. These two papers, the only two that support this resolution, so this Nida et al. Paper and the GoFlag data, these are the two plastid phylogenies that are essentially whole plastomes and have the largest taxon sampling. So that's very frustrating. Because it would have been really nice to say that, you know, if it was like two of the really old ones that supported, but no, we really have to consider this. We have to think carefully about what this means. I don't know what it means. I don't know what the answer is. So we have more thinking to do about this. I would say, to me, intuitively, this makes more sense. It doesn't make much sense to me that these two would go together. But there's clearly a signal from these plastid data that are suggesting that. So stay tuned as the fern community thinks about this more. Maybe Anna will figure that out for us, or Brenda will figure that out for us. So moving to the next one. So this situation up here, who is the sister group to the eupolypods, this tree, both of our trees actually have the same resolution, but it's been very controversial in the past, which is why I'm going to spend a moment talking about it. This one is not as sort of hurts your brain the way that the previous example does, because the dense steady ACE and the terrid ACE are both sort of morphologically reasonable looking ferns, and either would be a perfectly good candidate to be the sister to the eupolypods on sort of morphological grounds. These are the three resolutions that we see among all the data sets. So one option is that the dense steady ACE are the sister group. The other option is that the terrid ACE are the sister group, or the third option is that those two actually form a clade that is sister to the eupolypods. And so this is the distribution of the different studies, the different papers. So you can see that all of the nuclear data support this resolution with dense steady ACE sister to the eupolypods. And then the plastid data and mitochondrial data are sort of split among these three topologies. But these are the Go flag data sets. So obviously this is the one that I think is right. Yeah. And these are, as you can see, I mean, you can't even really see how it resolves because the branches are so very, very short. So it's not surprising that we have gotten a number of different stories over the years, but I think increasingly confidence would be in this resolution. So next we're going to jump down to the bottom of the tree, and I have these three groups highlighted in gray, but we're also going to talk about the Equisetaceae down here because they play an important role in this story. So this is another very long-standing, puzzling situation in not just fern biology, but sort of thinking about who the sister group is to the seed plants and thinking about the history of this part of the plant phylogeny in general. So I showed you all of these before, but just to remind you what they look like, the meradioids are tree-like. Psilotum, this is the group that has no true leaves or roots. The ophioglossoids have these sort of dimorphic leaves with separate sterile and fertile segments. And then the horsetails are the horsetails, very different morphologically from all of these groups. And if you talk to fern people will say to you with a lot of confidence that horsetails are sister to all the other ferns and that horsetails are ferns. And the reason that we say that is because we are thinking primarily of these studies that show this relationship with the Equisetaceae sister. So this is all of the ferns, all of the other ferns, right? So we've got seed plants down here, Equisetaceae sister to everything else. And these two resolutions differ in whether they put the Meradiales with Ophioglossalia and Xylitales or whether they put them together or not. And the Ophioglossalia and Xylitales always resolve together. So that's why I've shown them this way. So people who are most familiar with, sort of modern taxonomy and systematics and molecular-based studies will say with great confidence that Equisetum is a fern and it's sister to all the other ferns. Now, the paleo community does not feel the same way. The fossil record for Equisetum is complicated, very complex. Identifying homologies between structures is quite complicated. And we get, so Go flag, the data disagree. And then we also see these other four resolutions. Now notice there's like only one, in this case, 3 studies that support each of these other topologies. But notice how the horsetails bounce around in all of these 4 topologies. And in none of them, of course, are they the sister group. They're always embedded somewhere within these other eusporangiate lineages. And I didn't mention that earlier when I was introducing them, so the leptosporangiates, again, they have that specialized sporangium structure. All of these other groups, I highlighted these 3, the Meradiales, Ophioglossalia, and Xylitales as being eusporangiates, but the Equisetum, they are also eusporangiates. They have the same sort of structure, this eusporangiate structure. They're a bit different, the eusporangia among all these groups, but that's not like a character that would strongly support this resolution, one of these resolutions, for example. So this is a case where I think we still don't really know what's going on. The molecular data seem to be, the majority of these studies, including all of the nuclear studies, support the horsetails as sister. And then, of course, there's difference of opinion about these other lineages. But the fossils can't be ignored here. And that record, I think, needs to be considered much more than it has been. And there have not been that many, or perhaps any recent total evidence analyses that have tried to include fossil data in with the molecular data. So we clearly have more work to do to figure out this situation. And then the last one that I want to talk about is this one right here in the middle, that involves this set of families and orders. And this one is interesting because it has taxonomic implications, so naming implications. So the question here is how the Hymenophyllales, so these are the filmy ferns, very, very beautiful, morphologically interesting because they tend to be only one or a few cell layers thick. So they tend to live only in very wet, rainforest-like conditions, or at least very humid conditions. So how they are related to these three families, and whether these three families form an order or not. So they have historically been considered to be the order Glyciniales, but as you'll see, support for that order is equivocal. It's unclear whether we actually should recognize that order. And we are right now in the process of doing the second iteration of the Pteridophyte phylogeny group, our classification. And this is a big question to figure out whether or not we recognize the order. It was recognized in the previous classification, but that might have to change. So here are the first two topologies I'm going to show you are the two that have a monophyletic Glyciniales. So they put these three families together. And this is another situation where Metoniaceae, this is a small family. It only has about 4 species, and it's not included in many studies. But whenever it is included, it always goes with Dipteridaceae. And that sort of makes sense. You can, they have this very interesting sort of fan-like morphology. So morphologically, they're quite similar to each other. And anytime Metonia is included, it always goes with Dipteridaceae. So I've put them together. And then the Glyciniaceae, this is an interesting family because it has basically indeterminate growth, which is rare in ferns. So they're sort of scrambling and vining. So lots of an interesting morphology in that group as well. So you can see that from the plastid data sets, there's a lot of support for Glyciniales, for the order, with Hymenophyllales either sister to that clade plus all the other leptos, or actually forming a clade with the Glyciniales. But one way or another, support for that order. But then there are another 3 topologies present in these data sets that do not recover that order. And the stars here mean that the sampling in those 2 studies was such that we could not distinguish between those 2 because Glyciniaceae was missing, actually, which meant that those 2 studies would be, they could support either of these 2 resolutions. So most of our nuclear studies are supporting this down here. One of them is supporting the first nuclear study that was done, so supports this topology, but there's not a lot of support in the nuclear data for a monophyletic Glyciniales. I think that a proposal, you may remember, I actually don't remember, I think a proposal was submitted to the Pteridophyte phylogeny group as a community sourced thing. And the way that we're doing it in the second iteration is actually using GitHub. And people are proposing their taxonomic changes in GitHub, and then the whole community gets to vote. And I believe that there was a proposal put in based on this resolution to say we should not be recognizing Glyciniales, and I think it was rejected. I think the community really wants to cling to the order. And there's a lot of difference among all of these data sets. We're seeing a lot of different support. Again, a lot of different support from what we're seeing from the plastid data sets, including GoFlag, which is up there. That's the GoFlag plastid data compared to the majority of the nuclear studies. These branches are so short, and my opinion is that this is a real discordance. This might just be a place where the diversification was so short and so quick, and the nuclear genes did not have time to sort themselves. And we might just have to be unsatisfied with the answer, or maybe whole genomes is the answer. But fern genomes are enormous, as was alluded to earlier. Jim mentioned that. So whole genome-based phylogenies for ferns, I think are a ways off. So I don't have an answer. Moving on. So the last couple of slides, I just wanted to talk quickly about our molecular dating results. So what I'm showing you here are dated trees. These were done with TreePL. These data sets are so big that Bayesian approaches do not work. So we used TreePL to do this. So I'm showing you superimposed on top of each other the nuclear and plastid dated trees. And the calibrations that we used were from this paper, Schutpelz and Pryor, 2009. This was a beautiful paper that published, it was the first paper to publish a really comprehensive set of fern fossils with an appendix that's like dozens and dozens of pages long, explaining each fossil and why it was dated the way that it was. And that data set is still widely used for people to choose their fossil calibration points in fern phylogenies. So two things that I want to point out. I'm extremely impressed by how closely the crown groups match up. The ages of the crown groups are very similar between the nuclear and plastid trees. So, where you can barely tell the difference between those two. And this part of the tree, for example, too, the dates are very similar. But if you'll recall, there are some points of discordance between these two trees, and then that's also reflected in different dates. So the dates are obviously going to be a bit different where the topologies are different. So you see that in a few places as well. And the root is also quite different down here. And then, so those are the two studies, or those are our two data sets from GoFlag. And then if you put our molecular results, our molecular dating results in the context of all of the previous fern studies that have done molecular dating, it's all over the place. So what you're seeing here, each of these columns corresponds to a family. So here are all the families going from oldest roughly to most recent. These are the different studies by color. And then the data type, the triangles are plastid and the circles are nuclear. So these are the pink ones here, the plastid studies. The blue ones are nuclear studies. And then in green is GoFlag because we had both plastid and nuclear. And here you can see the GoFlag data with two separate sets of calibrations. So this study, this green color used the Schutpelz and Pryor calibrations. And so then those are the dates from the Schutpelz and Pryor paper. And then we also use the calibrations from this Nida et al. 2002 paper, which is a really nice paper where they've tried to do sort of an update to the Schutpelz and Pryor. And they've created a GitHub-based database of all of the confidently dated fern fossils that are known. So it's a really great resource. And so we wanted to see what our trees would look like if we use those calibrations. Interestingly, those dark pink, that study, their dates are some of the oldest. So then these are the stem ages, not the crown ages, the stem ages of all the families, ranging from very old to most recent. And so you can see that there is huge spread here, right? Like there's like 150 million years of spread in the ages of most of these families. So, molecular dating is about as imperfect a science, I think, as we have. So there's a lot of room for additional work and additional improvement. If you find the green ones in here, if you can spot where they are, they're pretty close to each other, as I noted in the previous slide. But then, yeah, if you look across all of these other studies, I think this is an area where we really have a lot of work to do. So if anyone says very confidently to you that a certain fern family is a certain age, you should laugh and tell them, ask them what study they're referring to. So with that, I want to acknowledge our funding came from the National Science Foundation from this Go Life, Go Flag grant. These folks were the PIs on this grant, and they were obviously instrumental in getting the funding. On the very first slide, there were 3 names at the bottom, and I should have mentioned on the first slide, and that was Gordon, Jesse Pelosi, who was one of my PhD students, and Susan Fawcett, who's an independent researcher now at the University of California, Berkeley, and they were instrumental in doing a lot of the analyses for this talk. And then these are all of the people who contributed fern data for GoFlag, but there were many, many other people who contributed gymnosperms and all the bryophytes and all those other groups as well. So really appreciate all of their contributions. And thank you again so much for the invitation to be here. Thank you for listening to the talk. And yeah, I'm happy to take questions. Thank you.
Now, Emily, thank you very much for this amazing talk. I think we will have a lot of questions, so I will, of course, immediately.
Thank you. Thank you for the talk. Would you indulge me to show me the plastic tree versus the nuclear tree?
Yeah, sure.
Yeah. No.
Oh, yeah, I
That
guess,
one.
yeah, any
Yeah,
of these
that
will
one.
work, yeah.
So what I see here, well, so you have the plastic tree with concatenated data, and then the nuclear tree is a species tree for an astral. And so definitely very different methodologies. But what I see is the nuclear tree has fairly long branches, like you mentioned, right? And worse resolution, I would dare to say, if I am not totally blind from here, than the plastic tree. So one question I have is why do you think that is? Of course, again, these are two very different methods. And then the second question I have is if you explore the effect of missing data. And I think that's important because sometimes the substitutional models, in order to accommodate the missing data, they have to add substitutions to a branch, right? So you end up with a lot of missing data that could become also row taxa, that could give you long branches that become row taxa, and they bounce around and you lose resolution. What do you think of that? And if you explore the effect of missing data?
So, okay, there's a lot there. So for thinking about the nuclear side, there was some missing data, but if you think about like the coverage within the individual gene trees, we actually did not have huge amounts of missing data for the individual gene trees actually had quite good coverage. And so what you're seeing here, you're not going to see because this tree is not based on looking at sequences, right? So you're not going to see a reflection of missing data in that way on the nuclear side. You would see that on the plastid side, right? Because the nuclear tree, this is a tree derived from trees. Yeah.
Say in these probes, right? You said, well, we have little missing data because a lot of my taxa amplify many of these thousands of loci, right? But then when you look closer, then for some taxa, you might get just a few base pairs of each loci, right? So it's not enough to look at. So I don't know what you mean by
But
coverage.
we didn't have that situation though. I agree that can happen, but we did not have that happen. Yeah. To your other, so the only points where the resolution differs is the points that are these black lines. So anywhere that it's blue along the backbone, for example, or yellow here, the resolution is the same. The lengths may differ, but the resolution is the same. So they're only different at this point here, and then these branches down here, which are those really short branches. So, I mean, I actually think overall, they're pretty congruent. And again, these places where they do differ are these places that historically have been very difficult because they're so short there. Yeah. So I'm not surprised by it. Yeah.
What was the proportion of missing data on each of those data sets? Might give you some insight.
I have a question.
Okay.
Oh, is it okay? Oh, then you. I was also curious about missing data in the branch lengths. Is this an astral 3 or an astral 4 tree? Because I know the branch length method is now substitutions per site, and before it was in coalescent units.
I can't remember, actually. This is in coalescent units, I think. Yeah, this one is in coalescent units.
I remember we had probably got a reviewer that said, oh, 3PL with coalescent units? No. And then we're so happy that the new version came out that estimates substitution per sites to get better clock estimates. Oh.
That's good. We'll keep that in mind for the dating analyses. Yeah.
Okay. I'm super interested in your talk. There are two particular things that I would like to bring up. One of them is the position of equisetum And I was a bit surprised to see earlier in the talk that you showed that in some cases it could go with seed plants. But
Yeah.
then looking at the summaries that you showed, it always seems to keep going with the ferns, which is the result from that Kathleen Pryor got in 2004. It has been.
Yeah. So so this is her 2004 paper. Earlier than that, she did get a result. The 2001 paper was the one that found it to be sister. So yeah. No, to all the rest of ferns. So she
Yeah.
published a paper in 2001 that was like the horsetails
yeah.
it was a big deal. It was in Nature, I think, or Science.
Yes.
But then this paper had greater sampling in 2004 and actually showed this resolution with Equisitales sister.
within.
Yeah, within sister
Okay.
the meradioids.
Actually, my impression from back then, of course, it depends to which paleobotanist you talk to, but some of them were kind of like okay with the result, maybe not with Equisitales within, but as sister to the rest of eusporangiate and leptosporangiate ferns.
I think
Because
that-
and even if you look at the paper by the book by Kenrick and Crane, Equisitales comes, and it's even before that the molecular studies came out.
I think that they object to it being called a fern.
Yeah,
I think they want it to be considered to be a separate thing.
it's fine, but they're the same clade.
Yeah.
So
Yeah.
so that was my first thing, and the other thing is looking at the dating. It's very clear that it's all over the place. I mean, I'm not surprised, and I completely agree with your comments about dating analyses. But I think, in summary, there's a paper that was that came out in 2004 that was entitled "Ferns Diversified in the Shadow of Angiosperms."
Yeah.
Is that still holding? And of course, ferns, it depends what you're talking about. But in that paper, they were talking about this clade. I don't know if it's I think it's the eupolypods that they're talking about, that they diversified after the evolution of angiosperms.
Yeah.
Because they were saying that what's happening is that angiosperms opened these whole niche for ferns to diversify. And it's the Hymenophyllales and things like that.
Yeah. So those groups start roughly over here someplace. They're not in sort of phylogenetic order, to the extent there is such a thing. They're in but those would be this suite of families over here. So the idea is that if you draw a line here, roughly 165 million years ago, we have angiosperms come on the scene. And that if you sort of draw that line there, that the majority of fern diversity appears after that point. And so this study, the Nida et al. Study, they called that into question because their dates for many of these families, which would be, again, roughly it's this side of the tree over here, this suite of families, do fall below that. But again, these are the stem ages. And so it's, I would say, still possible that the crown ages of, even with these old dates, could still have happened, that diversification happened in the shadow of angiosperms. So I think the hypothesis still holds up, regardless of which set of these dates ends up being correct. Yeah. No.
I have a question now. Well, first,
So in some of those, the plastome and the nodes in the plastome phylogeny are extremely short. Did you ever, either in the plastome analysis or in the astral analysis, I guess you looked at this, can you, with confidence, reject the possibility that you have polytomies?
No.
Oh, okay.
Let me think about that. So like any of these really short ones over here.
Yeah.
Yeah, I mean, it's possible. I mean, with the plastome, I would expect it to resolve one way or another. I mean, it's surprising ever that you would have this much lack of resolution. But again, the branches are really short. Yeah. I think that's really the only answer I have. I mean, and they are, again, thinking about ages and getting back to these, especially these down here, that would be potentially, 200, 300 million years, that we might be getting into, where there's been multiple substitutions at a site, maybe. So that could be playing a role there as well.
Can I ask one more nitpicky, or not
Of course.
nitpicky, but specific question? So down with the Salutaceae, Ophicolaceae, and Narutaceae, in the gene trees don't show equal frequency of the two alternatives. So with your tree with your where you've got the high diagrams.
Oh, yeah, all the way back at the beginning.
Yeah, sorry.
No, that's fine.
And I'm wondering if the plastome is
There we go.
looking like, yeah, as I recall. So yeah,
Yeah,
purple is much
yeah.
larger than
Yeah, even
red.
these nodes even
Yeah.
here, yeah.
Yeah.
There's more black rose. So
So I wonder if there's some, something else going on there than besides just coalescence being responsible for those conflicts, and whether or not there could have been plastome capture down there.
that's true. That would complicate things enormously, but it's possible. Yeah. I mean, there are within the nuclear genome, we know about horizontal transfer events, but plastome capture, I don't know of any examples of that in
Well, just by some moving, being captured, but through hybridization.
Yeah.
Might be-
Yeah, that's a good point. And certainly possible given how prone to hybridization and polyploidy ferns are. Or that could have been something. I mean, all of these problematic nodes potentially that could be at play. Yeah. Yeah, that's a good point. We should look at that more. Yeah, we have a little chunk of, yeah. Yeah, and this one, there's there too, yeah. Yeah.
Dave?
Last question for me. We love morphology, and we read a lot of papers about how great it is to include some morphology when you have some of these, difficult to resolve branches. And are there plans to include morphology soon with this kind of stuff? And how will you use that to try to answer these questions? Will you, for instance, concatenate data?
Yeah, we don't have plans to add morphological data at this point because the data, so I mean, I'm showing this nicely collapsed into families, but remember there's 1,000 species here. So I mean, we could choose a subset from each of these large groups, but that would be challenging, like eupolypods up here to really accurately capture their morphological diversity would be a big matrix, and even some of these groups down here, like Ophioglossacea, they're very variable. And one of the things that's frustrating is that there are so many unique derived features, like the tree ferns. Okay, we could add a bunch of characters related to the fact that they're tree ferns. That doesn't help us, right? Salviniales, the water, like we could add a bunch of characters for water ferns. That's not going to help us sort this out. So that's, I think, the challenge is that many of these groups where there are these short branches. I mean, when you get to the eupolypods, you could easily build a morphological matrix for that group, and probably for this whole group here, Densitaceae, Pteridaceae, and the eupolypods. But all of this stuff down here, they're so different from each other. And if I maybe let me go back a little bit to emphasize or to illustrate what I'm trying to say. I'm going to have to go back quite a bit. Oh God, there's a lot of clicking. Sorry. More clicking. What I want is almost there. That's what I want. So the eupolypods are this entire group down here. So that's the Pteridaceae, that's the Densitaceae. So all of what I'm showing you in those other trees is all of this collapsing this. But this, if you were able to see those very small numbers, this is where the majority of fern diversity is. And you could, but despite that, they're similar enough to each other once you get to the eupolypods that you could build a matrix pretty easily with a relatively reasonable number of characters. these things are all so different from each other, and they have all have unique aspects to them that trying to use morphology would be extremely difficult. And it's why it's been so difficult. Yeah. So that's not to say we shouldn't try to use morphology. Of course we should. And fossils especially would be wonderful to include more than we have. And nobody has done like fossilized birth death process with ferns. Nobody's done that. So there's a lot of open ground there to explore. So yeah, lots more work to be done, which is great.
So I will take the last question. Have you tried to mine mitochondrial data out of the GoFlag data?
We did try, and it wasn't, we didn't try very hard. And we weren't super successful. The chloroplast was much easier. The mitochondrial, I think there's a reason there's only one mitochondrial paper in ferns. There have been a few others that have looked at mitochondrial structure, but again, the mitochondrial genome is weird always, and it varies a lot across all these different groups. So we were struggling with it, and I think probably for that reason.
Yeah.
So we could try harder, and it would definitely be worth further exploration of the mitochondrial genome for sure.
Then would you expect additional discordance if you finally have a mitochondrial?
Probably.
Okay.
I mean, I think for the most part it matches, but actually I don't want to say that. In some cases it matches with the plastid data, but not always with the chloroplast data, but not always. Sometimes it agrees with the nuclear. So yeah, I mean, it's potentially telling its own story. So yeah. So
Thank you.
come study ferns, because there's so much still to be done.
Thank you very much, Emily.
you.
So I will just give the word to Susanna so we can close this event. Thank you.
No, I just want to thank all of you for being here, for your presence, for the questions that you have made. And just remind you, I mean, these talks are intended for our community. You selected the speakers, and we hope we can continue to do this seminar in the following years, but more like an invitation for you to tell us who do you want us to invite, and we will do as much as we can to bring them. And I would just like to finish by thanking very much the Coordinación de la Investigación Científica for providing part of the funding, and the graduate program in biological sciences, el posgrado en ciencias biológicas, for providing another. And there are many people that I would like to thank, but I would really like to ask you and me to give a very loud thanks to Carolina Granados, Lázaro Guevara, and Ulises Rosas, who is not with us anymore because he went to take Mike Buruganan to the airport. But anyway, we're very thankful to all of them, and thank you very, very much. There's coffee out here.